Good morning, everyone. Today, my team and I will be talking about the scientific revolution of genes in terms of the shift from genetics to epigenetics. Firstly, I would like to start by describing the two terms we will be discussing about today. Genetics is the study of the function and composition of single genes, and it was first introduced into the scientific research world in the beginning of the 20th century. As the name implies, it has to do with genes, which are the specific sequence of DNA that codes for a functional molecule. Then we have epigenetics. The term of epigenetics and its use came a little bit later than genetics, in the beginning of the 21st century, and it's the study of the environmental changes affecting genes activity. Epigenome is a set of chemical modifications of proteins, altering the gene expression, as already explained by the term. Now you may will be wondering why we're bringing this topic to today's PowerPoint. And this is because it has been seen throughout the years of research that scientists had a shift from genetics, so the old paradigm, to epigenetics. And in this manner, the concept of paradigm shift was introduced, which talks about the fundamental change in the basic concept and experimental practices of a scientific discipline. The concept of paradigm shift was firstly introduced and explained by the American physicist, the Trinity and philosopher Thomas Kuhn, who was born in 1922. Kuhn believed and explained through his publications that there is a categorical change in internal parts of the previously establishing views of science. This metamorphosis can be summed up by the two major concepts that I would like to talk about. First, Kuhn sustained that scientific progress was in fact not a cumulative process, but it's rather characterized by periods of normal research interrupted by periods of evolutionary upheavals. This search for new paradigms is rooted in the willingness to try anything once the previous paradigm has been found insufficient. Secondly, Kuhn questioned the previously established notions of scientific truth. Where other scientists saw objective truth, he merely saw a consensus between the scientific community. Objective conclusions are based on subjective conditioning of the scientist, affecting the scientific truth and even the choice of which paradigm is going to follow. This theory altered the scientific world, shifting from a common sense-based view towards one that incorporates subjective notions. As shown in the slide, we can also have a look on the circle that Thomas Kuhn introduced that leads from normal science to the new paradigm. And it's mainly affected by the further information and facts that we have in regards to the topic that it's discussed. Now, I would like to dive into a more concrete idea of how the implications of Kuhn are demonstrated within our example, which is nothing else but genetics and epigenetics. So first, as already explained, the paradigm shift within genetics and epigenetics is rather obvious. The establishment of more facts about genes and the necessity to study them in more depth led to the shift of genetics to epigenetics. Second of all, we have the theory of Leibniz. In regards to that, it is believed that different perspectives of what is needed when studying genes caused scientists to express their preferences between genetics and epigenetics, and therefore led to the fact that some people were looking at the same concept, which is genes, in a different manner based on their belief, background, and the goal of their research. Last but not least, I would like to talk about incommensurability, which indicates that although genetics and epigenetics seem to be somehow correlated with each other, because of course they're both talking about genetic coding, in reality, they work on completely diverse aspects and thereby are considered incommensurable because of the fact that they study two completely different things and they approach the situation in two diverse manners. However, we should also not forget to talk about the fact that the shift from the old paradigm to the new one does not necessarily mean that the old paradigm is completely replaced by the new one. This means that genetics and epigenetics, although an old and a new paradigm, are still widely used in, let's say, the same kind of quantity based on what the scientists are looking for in the research. So genetic is used for screening in genetic diseases in generations, mapping disease genes, and diagnosis and treatment. Whereas epigenetics is about prevention of sporadic cancers, screening of non-editing disorders, medical implications, as well as better understanding of non-mutational diseases. As a result, it is widely explained now that the recent rise of epigenetics is not, in the sense of Thomas Kuhn's, a revolution, because the genetic modification that has been taking place did not replace genetic and genomic research, but just open up a new areas of research related to the old question, so as to find cures for a plethora of diseases and as well as to fight resistance to those diseases. All in all, we can conclude that even though such an example is not completely a pure form of revolution, as in terms of Thomas Kuhn's concept, that doesn't mean that it doesn't remain a solid example of paradigm shift. Now, my colleague Giovanni is going to continue with the relation of our topic to biomedical sciences.
Let's move on to more concrete examples of this paradigm shift, which we have encountered throughout the last 2 years. It is clear that many questions arising under the genetics umbrella of knowledge could not be answered by only applying genetic techniques. One of the first landmarks that introduced the new epigenetics paradigm happened thanks to the theory explaining how the body, the second X chromosome copy, was formed by a Hyston modifications. This scientific breakthrough led to an array of other scientific discoveries which made the soil fertile for setting a new whole series of plausible theories explaining nature's secrets. For example, scientists observed that the barrel body would disappear in breast cancer cells, thus hinting that epigenetics could also be involved in tumor growth, and ultimately this opened up a new world of tumor biomarkers. Another example of this paradigm shift was when the association of epigenetics and obesity was found. Back in the days, scientists were trying to give an explanation of the reason why the obesity phenomena was skyrocketing. And geneticists were finding genes involved with this disease, but it was not giving the whole picture of the story. When epigenetics was introduced, they actually understood that even dieting, so what we eat, can affect our gene expression without mutating the DNA. And these epigenetics effects can be passed down to the offspring. So we can see how even though genetic and epigenetics they share the same research field and they have a common language, they lead to different theories and different view of the world. So now let's move on to the ethical concerns and the recommendations that because these experimental practices need. And firstly, there are some ethical objections that were being discussed, and especially because scientists should not use these informations for their only individual purpose, but they should use it as a common scientific purpose. So these arise some privacy issues, as well as there are some issues for reproduction, because when we test and we work on the genes, this could lead to affect the offspring and affects of future generations. So we need to be careful on working on those techniques. And as well as there are some safety issues because there are on the long terms have not been demonstrated yet and experimented. So we need to be careful. And then also there's the possibility of psychological harm and which can lead to stigmatization of the individual with leading to health issues. And so the ethical committee discussed some guidelines that in order to avoid these problems. And firstly, clinical testing and trials should be all voluntary. So the patients had to be willingly willingly decide whether to take part of these clinical trials, as well as a genetic and epigenetic screening should be preceded by adequate information about the purpose and the possible outcomes. And also, in order to solve the privacy issues, the screening should be anonymous in order to so they can be conducted after notification of the population, and ultimately the results should not be disclosed. So this is the final recommendations, and these are the resources. And thank you very much for listening to us.